So this video is about molecular phylogeny, otherwise known as cladistics. There's a few syllabus objectives here. First of all, we need to be able to describe the classification systems for molecular sequences called molecular phylogeny, otherwise called cladistics. So it's describe this type of classification system. We need to define the term clade, and we also need to be able to recall the common assumptions of cladistics and stating what these are. Okay, so first of all, what is a molecular sequence? Well, so it's the order or the sequence of different molecules, either DNA nucleotides, your A's and your C's and your T's and your G's, or the sequence of amino acids, which of course are as a result of the DNA nucleotide sequence. So it's a process of actually having a look at what that sequence is, and it then is able to be used as a comparison between the genetic sequences of different organisms to be able to look at how they're related from an evolutionary point of view. The assumption is that the more similar the genetic sequences are, the more closely related they are, and the more different the molecular sequences, the more distantly related they are. So here's an example of a molecular sequence. It's certainly not A's and G's and T's and C's, so it's not DNA. Perhaps it's amino acids, but the point is it's a sequence. And you can see that we're looking at comparing different species here. And if we're having a look, we would assume that the human and the chimp are most closely related. And you see all of these ones that are in grey across all of these species are the same. All of these sequences are the same. The ones that are light in colour are the ones that are different. So we're able to compare those differences. And you can see if we run all the way across, humans and chimps have the same sequence for all of these. Whereas when we have a look at some of these other organisms, you see that they're different. So from this sequence, we can hypothesize a specific evolutionary relationship. And we can represent it graphically like this. So those sequences we saw that were the same, this is what we call highly conserved sequences. It's the DNA sequences that are the same or similar between species. They have been maintained or conserved by natural selection. In other words, the sequence is important enough that it provides some sort of evolutionary advantage to the species, so therefore there's no need for it to change through evolution, so it tends to be conserved. It's helpful to have these conserved sequences because what it allows the scientists to do is actually line up the different sequences based on the conserved sequence, and that way they can then look at identifying the differences with a side-by-side comparison. So there's a number of different species here that are related to different moths and mosquitoes, and they've lined them up based on this sequence here of a highly conserved sequence, and they're able to identify where the insertion of the mosquito mutations occurred, and then also the insertion of the moth mutations. So if that's molecular sequencing, cladistics or molecular phylogeny is the process of comparing these similarities and differences in the genetic sequences of organisms to determine that evolutionary relationship. Now, it makes several assumptions. The first one is that it assumes that organisms with similar sequences are closely related. It also assumes that there's a common ancestor. Now, a common ancestor is the most recent species from which two or more different species are descended. So we could represent a common ancestor here, and from that we have a split, and we have one group of species go in this direction, one species group go this way. You can understand that time goes in this direction here along the x-axis, and this is a measure of time. It doesn't have any units, but it's a measure of time, and we're talking about geological time here. Evolution obviously takes time. It's very slow. But so if we think about a common ancestor is, with this representation, it's this horizontal line here where we've got a split or a bifurcation, and then we've got two different species. Okay, so the common ancestor is here. The most recent species from which two or more different species are descended. So that would be a common ancestor for these two descendants. This one here would be the common ancestor for these three species. So there's several common assumptions of cladistics. Firstly, common ancestry. So species are derived from a common ancestor. If we trace it back, we can find a point where they share a common ancestor. The next one is that we have bifurcation. So bifurcation means this split, and this occurs when there's a mutation, a change to the genetic sequence, and that is where these new species derive. You know, every one of these is a bifurcation. Also, we describe these as nodes. N-O-D-E-S. Nodes are the bifurcations, and they're the splits. And the third assumption is that we have change over time. And in fact, we can actually use this as a bit of a molecular clock. And so this is the guidance material in the syllabus. So it's important for us to understand this. Conserved sequences. Now these occur in DNA, so nucleic DNA, but also mitochondrial DNA. So conserved sequences, we assume that mutations, so changes in the DNA sequence, occur over time, and that is at a constant rate. So therefore we're able to count back the number of changes or number of mutations to be able to estimate the length of time since there's been a common ancestor by the number of mutations in geological time. So based on that, we can essentially create a molecular clock to be able to calculate the degree of divergence based on the number of mutations that have occurred. Now, one way that we can actually represent these phylogenetic relationships is through a branching diagram called a cladogram. Now, we're going to find there's lots of different types of cladograms, and in a future video I'm going to show you how to interpret cladograms. Basically, a cladogram demonstrates those assumptions of cladistics. So common ancestor, bifurcation, change over time. In both of these, change is essentially going in a vertical direction, but it depends on how they're orientated. Okay, so common ancestor. This would be a common ancestor, and this would be a common ancestor for A and B. Another thing we can represent on these diagrams is what we call a clade. This is a specific syllabus objective to be able to define clade. This is the definition of clade provided by the syllabus. It's a group of organisms that consists of a common ancestor and all of its lineal descendants. So that is a clade, a common ancestor and its descendants. But that is also a clade, common ancestor and its descendants. That's a clade, and that's a clade. What's not a clade though? We wouldn't say that A and C are a clade. We wouldn't say that B and C are a clade. Okay, it's got to be all of its descendants and the common ancestor. So to finish off today's video, here's a question from one of the external exams, a multiple choice question. So this question asks you to determine, is this the correct sequence?